Hi everyone. Today we will be talking about different types of fermenters. We have already discussed what is a fermenter, what are the parts of fermenter and its functions. The topic is under the paper Industrial Microbiology. Okay. From this video you will get a quick overview on few types of fermenters. Okay. Before starting with different types of fermenters, let's see what is fermenter. Fermenter is a vessel in which sterile media and microbial culture are mixed and fermentation process is carried out. It provides a sterile environment and optimum condition for the growth of microorganism and the synthesis of new product. Fermenter is made up of stainless steel and it is also called bioreactor. There are different types of fermenter: batch fermenter, continuous fermenter, fed batch fermenter, liquid state fermenter, solid state fermenter, tubular fermenter, membrane bioreactor, photobioreactor, packed tower bioreactor, bubble column fermenter, airlift fermenter, stirred tank fermenter, and fluidized bed fermenter. Fermentation can be carried out as batch, fed batch, or continuous. In this video, we will discuss about this type of fermentation or this type of fermenters. Okay. The choice of the mode of the operation, that is batch, continuous or fed batch, depends on the product being produced. Let us look into the figure. The first image indicates batch fermenter and second one indicates fed batch and third one indicates continuous fermenter. Batch fermenter is a closed system. Fed batch is a semi-closed system and continuous fermenter is a open system. In batch fermentation, we provide nutrient medium, microorganism and everything at the starting time and collect the product at the end of the process. In the case of fed batch fermentation, we provide little amount of substrate at several times and collect the product at the end of the process. Okay, so the addition of substrate is required at several times. Okay, so the addition increases the volume of the media. Okay, here the volume is not constant. Okay, in the case of batch fermenter, the volume of the media is same, that is constant. Okay, but in the case of continuous fermentation, we provide the substrate continuously and collect the product many times. Okay, so the addition of media require many times and the collection of product require many times. Okay, so it is an open system. First, we can discuss batch fermenter or batch fermentation. Batch fermenters are used to carry out microbiological process on a batch basis. Batch fermentation is regarded as a closed system. It is a process in which a large volume of nutrient medium is inoculated for the harvest and recovery of the product. Okay. At the end of the fermentation, the vessel or the fermenter is cleaned and re-sterilized for the subsequent batch. It is a closed system in which all the nutrients are added initially. Okay. In batch fermentation, the growth of the microorganism follows the characteristics of the growth curve. That is, initial lag phase followed by log phase, finally reaching a stationary phase. The stationary phase attains due to nutrient depletion. Batch fermenters are used to carry out microbiological process. The capacity of the batch fermenter ranges from few hundred to several thousand gallons. Small laboratory fermenter, pilot plant fermenter, and large fermenters are available. For small lab fermenter, 1 to 2 liter to 12 to 15 liter capacity. In the case of pilot plant, 25 to 100 gallons to 2000 gallons. In the case of large fermenter, 5000 to 10,000 gallons. Okay. A headspace is located at the top of the fermenter. This space is to allow aeration, splashing, and forming of aqueous medium. Nutrient medium inoculated with microorganism. It may be necessary to add acid, alkali, or antiform agents. Okay. We can add acid or alkali or antiform agents during the processing time. But there is no addition of nutrient media or microbial culture. In the batch fermenter, the growth of the microorganism follows the characteristics of the growth curve. That is, 6 phases of growth can be seen in batch fermentation that are listed here. First one, lag phase. Second one, acceleration phase. Third one, logarithmic phase. Fourth one, deceleration phase. Fifth, stationary phase. And finally, death phase. You have already studied growth curve and its significance. Okay. Here in this figure, the 6 phases of the growth curve can be seen, or the 6 phases of the microbial cell can be seen. Okay. Lag phase, log phase, stationary phase, and death phase are marked in this graphical representation. Acceleration phase and deceleration phase are indicated by A and B. You can see the characteristics or the significance of the each phase. First one, lag phase. It is the initial period of culturing. Okay. In this phase, microorganisms take time to adjust new environment. Hence, the lag phase is also called phase of adjustment or time of adaptation. Second phase is the acceleration phase. It is a brief period in which the cells starts to grow slowly. Acceleration phase connects log and lag phase. Okay. Then next phase, log phase. It is also called logarithmic phase or exponential phase. In this phase, microorganisms grow and multiply. Cells grow at maximum rate. Okay. Thereby the cell mass increases. A straight line is obtained in the log phase. Next phase is the deceleration phase. Here the growth of the microorganism decreases. Then they enter the deceleration phase. Then fifth phase is the stationary phase. The microbial growth slow down or completely stop in this phase. It is due to the nutrient depletion and the accumulation of toxic products in the media. The biomass remain constant during this stationary phase. Next is the death phase. Metabolic activity stops in this phase. This is due to the depletion of energy reserves. The cells die at an exponential phase. Okay. In the industrial fermentation, the growth of the microorganism is halted at the end of the log phase or just before the death phase begins and the cells are harvested. Batch fermenter contain pH control, temperature control, agitation. Okay. pH control is by acid or alkali addition. The addition is controlled by auto titrator. Okay. The auto titrator is connected to a pH probe. Okay. So the pH probe connects with the auto titrator and the auto titrator control the addition of acid or alkali. Thereby we can maintain the pH in the fermenter. Then next temperature control. It is by water jacket around the vessel. This is supplemented by use of internal coils to provide sufficient heat transfer surface. Third one, agitation. It consists of strong and straight shaft to which impellers are fitted. Impeller consists of blades. Different types of blades are available. The shaft passes through the lid of the fermenter and it is rotated with the help of a motor. Okay. Next is the time. The time required for a batch fermenter varies from hours to weeks. Throughout this time, contamination must be avoided and the vessel contents must be agitated for their temperature controlled. Second type of the fermenter is the continuous fermenter. It is an open system. It involves the removal of culture medium continuously and replacement of this with fresh medium in a bioreactor. Both the addition and removal are done at the same time or same rate. Okay, so that the working volume remains constant. That is, it is in a steady state condition. Addition and removal done at the same rate. Okay. In this type, the culture solution is homogeneously mixed. The contents of the vessel are at a steady state. The fresh medium is continuously added and an equal volume of the spent fermentation broth and cells are removed. Exponential growth will be prolonged. New biomass is balanced by the loss of cells. The flow of medium into the vessel is related to the volume of the vessel. That is, we can calculate dilution rate, that is indicated by D, is equal to F by V. Dilution rate D is equal to F by V. F stands for flow rate and V stands for volume of the vessel. Okay. Continuous fermenter or the continuous bioreactors are of two types: chemostat bioreactor and turbidostat bioreactor. Chemostat means substrate concentration is kept constant. The concentration of any of the substrate is adjusted to control the cell growth and maintain a steady state. Then the second type, turbidostat, means biomass concentration is kept constant. Okay. In the case of chemostat, substrate concentration kept constant. In the case of turbidostat, biomass concentration is kept constant. The turbidity measurement is used to monitor the biomass concentration. The rate of addition of nutrient solution can be adjusted to maintain a constant cell growth. Next, we can discuss application, advantage, and disadvantages of continuous fermenter. First, applications. It is used for the production of antibiotics, organic solvents, SCP, beer, and ethanol. Then the advantages. First, size of the bioreactor are small compared to the batch fermentation, and the yield of the product is more consistent because the physiological state of the cell is uniform. Then cost effective. Then the downtime between the two successive fermentations for cleaning and preparing the bioreactor for reuse is avoided in continuous fermentation. Okay. So we can save the time required for cleaning. Continuous fermenter have certain disadvantages. Okay. Continuous fermentation may run continuously for a period of 500 to 1000 hours. Maintenance of sterile condition for such a long period is difficult. It is not easy to maintain the same quality of the culture medium for all additions. So this is about continuous fermenter or continuous fermentation. Third one, fed batch fermenter. It is also called semi-closed system of cultivation, and it is used for the production of recombinant protein. Fed batch fermentation require more careful monitoring, so it is not preferred for industrial purpose. Fed batch fermenter used in the biotechnological process where one or more nutrients or substrates are added to the fermenter during cultivation and in which the product remain in the bioreactor until the end of the process. Here the base medium supports the initial cell culture and a feed medium is added to prevent nutrient depletion. Fed batch fermenter or the fed batch fermentation is an modified version of batch fermenter. In the case of fed batch, the substrate is introduced into the fermenter at different time intervals during the processing time. But in the case of batch fermenter, the substrate is added only once. Addition of substrate into the fermenter helps in the extension of logarithmic phase and stationary phase. This prolongation of the stationary phase increases the production of desired product. That is, most of the products are synthesized in the stationary phase. So the substrate addition increases the length of the stationary phase and thereby increases the product formation. Please draw this figure. In the fed batch fermentation, substrate concentration cannot be measured directly because it contains many different components. So other parameters related to substrate concentration are measured. Example, formation of organic acid, change in pH, production of carbon dioxide, etc. Based on the amount of substrate consumed, we add the substrate. One of the disadvantages of the fed batch fermenter is the contamination problem. So this is about fed batch fermenter. So today we have discussed first three types of fermenters: batch fermenter, fed batch fermenter, and continuous fermenter. We will discuss other types of fermenters in the next video. Okay, thank you.